There may be a few more people coming into the room, but it's my pleasure to introduce the third speaker of our session today, Christopher Penfold from the University of Warwick, who is going to talk to us about nonparametric Bayesian inference and perturbed and orthologous gene regulatory networks. Christopher?
Okay, can everyone hear me okay? Yeah?
Everyone here?
I guess that means yes, people would shout if not. So just to avoid disappointment, today I'm mainly going to be talking about these perturbed gene regulatory networks rather than the orthologous gene regulatory networks. Now, this is not because the methods can't necessarily be applied to orthologous networks. It's just that on the project I'm working for, we don't tend to have this kind of data, but we do have this in large quantities. So this slide more or less summarizes what I'm interested in, and this, I guess it's an oversimplification really, so we're missing all sorts of regulatory controls, so we don't have any epigenetic effects here. But at the same time, I think it's still quite important in regulating how an organism works. So here you have your gene, and your genes can be transcribed into mRNA, and then this mRNA can be translated into proteins. Now some of these proteins are involved in changing the metabolites, and others can form complexes, etc., or bind on their own to the gene in the promoter regions of the gene. And in this way, they kind of influence the expression of that gene. So what you can get out of this is this network here, this projected network, the dashed lines of a gene regulating network. And this is what I'm interested in trying to identify. So this is going to be fairly important for a number of reasons. So truly understanding this and all these as well is quite important for kind of really understanding how an organism responds to the environment. And this is going to be very important when you want to predict how it might respond in novel environments. So for example, in light of climate change, how will our crops respond to changes in temperature, changes in various other factors. And at the same time, really understanding these networks will hopefully allow us to make really targeted genetic interventions. So again, going back to this climate change idea, if we don't want to necessarily start growing another crop, can we make the particular crop more responsive, less influenced by stress? So there are a number of ways of identifying these gene regulatory networks. On the one hand, you have your experimental methods for identifying regulation directly. So you can have chip or chip sequencing, and you can also have various other things like yeast-one-hybrids, bacterial one-hybrids, and a variety of other methods for identifying regulation. These tend not to be so high throughput, but I guess we are gradually getting there, and we're potentially getting to a stage now where we can maybe just measure fairly accurately what the networks are. At the other end of the scale, we might want to take this kind of systems level approach where we measure everything in terms of the transcriptome and potentially protein levels as well, and try to infer these networks from time series measurements. Now, one of the things I'm particularly interested in to do with these networks is what happens when you have two different conditions. Can we start to identify similarities and differences in the networks under certain conditions? So here's for example, is maybe a drought-induced senescence where the leaves start to yellow, and this is Arabidopsis infected with a pathogen. So are there similarities and differences in these networks, and can we identify them? Now the other aspect I want to talk about is, can we leverage multiple types of different data? So here is an example of our Yeast-one-hybrid data set, and already it's quite dense. So this indicates whether a transcription factor can bind the promoter region of a particular gene. So this is like an experimentally measured network. And so can we start leveraging this alongside our time series data to see what parts of this are more accurate and what parts are perhaps not? Because the caveat here is that these kind of networks are noisy as well. So it's not just the time series data that's noisy. This can also be very noisy. And on the side note, it's especially noisy when they let me into the lab. So here you've got all sorts of pipettes dropped, and you got the yeast having to form some sort of defensive line against whatever this is. So the methods we use to try and identify these networks is based around a causal structure identification. I've gone too far. based around a causal structure identification algorithm, which was originally suggested by Sandy Clem, who's now at MIT. And there are some other related methods here. So the idea behind this is that you have a dynamical model of gene expression. So this is a discrete time nonlinear dynamical system. And there will be a continuous, so there's an equivalent continuous version of this would be, for example, an ODE. So that would be going from discrete time to continuous time. And what we're actually interested in is measuring just a particular component of this, or predicting a particular component. So here we want to see how the JAPE gene evolves as a function of the gene expression at the previous time point and this causal structure, or in this particular context, the parents or the regulators. We also have some noise on this. So if we already know what the parents are, or the regulators are, what can we say about F in this case? So the first thing we might want to do is have a look at what the data looks like. Well, for the JAPE component, so gene J, you have a vector of observations running from time point T1 to Tn. And we're modeling that as a series of inputs for all the parent genes. So in this case, we've just assumed there are G genes. And this input runs from time 0 to time point n minus 1. So essentially what we want to do here is just find a function that maps this input matrix to this vector of observations. So one of the ways we can so this problem is encountered quite a lot in machine learning, and one of the ways to approach it is to use Gaussian processes. So a Gaussian process is a way of placing a prior distribution over functions. So I'm trying to illustrate that here. So here we have a zero mean Gaussian process, and we've just drawn samples from this. In the same way that you can draw samples from any other distribution, you can draw samples, and what you get is functions. And at the same time, if you have some data, you can update this via Bayes' rule, and you can draw functions from the posterior distribution. So a Gaussian process is fully defined by its mean function and covariance function. I don't really have time to go into it too much, but if people are interested in learning more about it, there's a nice book by Rasmussen and Williams. Now the key points here are that you can make predictions at new positions, so you can begin to predict how this system might respond at new points. And the other thing is that we can identify marginal likelihood, so we can marginalize over the functions and just calculate likelihood, the marginal likelihood for the observation, give the input, and our latent variable, the parents. Now this is dependent upon the choice of covariance function, so you have to specify that. And generally, people tend to use the squared exponential covariance function. There are a variety of others, and this, so there are loads of covariance functions you can use, but this tends to be the most used one. So the trouble is that the regulators are not always known ahead of time, and we must learn these. So again, we can try and do this via Bayes' rule. So here we've just got our Gaussian process likelihood, we've got some prior distribution here, and we've got a normalization constant. Now to evaluate this, we have to evaluate a Gaussian process model over all combinations of parents. So this it scales quite horribly, it scales factorially. So one of the ways you can address that is you can limit the number of transcription factors that you expect to bind simultaneously, and then you're normalizing over, so this summation takes place over a much smaller set. So this is a graphical representation of the inference that we want to do. So it's just another way of summarizing the model. And so here we have our observation data for gene I in this case. And we also, this depends upon the expression of the previous time points, plus this latent variable and some parameters in the Gaussian process model. But if you recall, what we actually want to do is we want to look at gene networks in cases where there are different treatments, where the network itself might potentially be different. So naively, you could do something along these lines where you just have an inference for each of your data sets. Now the trouble there is that you tend to be throwing away mutual information. So for these two networks, this example, there are a lot of similar connections, a lot of identical connections in these two networks, and it's only a few cases where you've got something missing here, which is in there, and an extra regulation here that are different. So we have gone from this kind of modeling to something like this. So here you can see your standard CSI model I've just spoken about. And in this case, you've got one of those for each of your data sets, and you've got some constraint over what these parents can be. So you can write out posterior distribution for all the parameters in your model. So in this case, your parameters are your regulators of your gene in each data set, your hyperregulators or your hypernetwork, some parameters for the Gaussian process model, and you also have some additional parameters. So this can be broken down into various things. So this is just the prior distribution over the Gaussian process parameters and some other parameters. And those tend to be positive, so we have an independent gamma prior. And then you have something like a prior distribution over your hyperparents. So you know that is typically something like a uniform prior, maybe. So now we look into the product, look at the terms in this product. So here, this just captures the dynamics of the data set. So it's probability of your observation given your input and your parent. So again, this is just the Gaussian process model. We can evaluate this quite nicely. And we also have a constraint on the topology. Now, in this case, we've set this as a Gibbs distribution. So basically, the probability of a particular set of parents given a hyperparent net is just related to how different those networks are with some temperature parameter. Now, this temperature parameter controls how much influence the hyper network has on the networks in each data set. So if this tends to 0, you essentially allow the network in a data set to be independent of the hyper network. Whereas if this tends to a very high number, you're essentially enforcing the network in a particular data set is identical to your hyper network. So we've coded this up and we've run this on various in silico data sets, and we found it gives fairly nice results. And within the model, this constrained hypernetwork can allow propagation of information between data sets. And at the other end of the scale, if these data sets have independent networks, you don't really lose out. So you're not propagating bad information through. And that's because these beta parameters can allow you to tune how much information is propagated. So now we come to what we actually want to do. So can we identify it? So going back to our Yeast-one-hybrid network, can we identify a stress-specific response? So in this, can we pull out subnetworks that might be lit up or set going in a particular stress? So in this case, because we already have a putative network, we can set the hypernetwork in this case to whatever that network is. So in this case, you might say it to the case where all of your putative regulators are binding simultaneously. And this represents a kind of weak prior towards everything binding in every single stress. So we've run this on a gene. So we found a gene called RD29A, and this is implicated in plant cold stress response. And it's not really implicated in much else. And so we also, so we have the putative regulators here. So these were picked up by Yeast-one-hybrids, and there are a couple of other things that were picked up by microarray knockouts, etc. And then we have various data sets here. So recall that this is picked up, so this is implicated in cold response. So we can look in each data set whether a particular regulator is binding. Now, I think the key point here is that we've set the hypernetwork as a case where everything's binding simultaneously. So if there's no information inherent in the data, the best it can do is go to whatever the prior information is. And so in this case, it's going towards everything binding simultaneously. Now, on the other hand, if there is a lot of information in the data, you can really tune a specific network. And so you can see here, by and large, most of these tend to be red, kind of reddish or quite high blocks. So these are all relatively uninformative data sets. And if you look at the profiles, they're relatively uninformative, and a lot of them are either not differentially expressed or they're not expressed at all. So there are two cases here. So one's the cold, where you really get a signal coming out. So you get SEDG1 and some of the DREBs coming out with slightly higher probabilities than all of these. And then you also get some lower probabilities in these osmotic data sets. So what this does is this allows you to start formulating hypotheses of where you might get switches. So in this case, these genes are probably not involved in the cold response, but they are potentially involved in the osmotic response. So we've essentially tagged some extra interaction onto a pre-existing network. So this cold stress response, this section here, is available in the literature, and we've identified potential switch where osmotic stress brings in other regulators. So finally, in summary, we've used the method CSI, which is a method for inferring sparse networks. Now, I kind of want to question whether gene regulatory networks are really that sparse, because you can see that there are a lot of things potentially binding in the Yeast-one-hybrid. And I would argue that they are potentially still relatively sparse. It's just that you have different subsets binding in different conditions. And so this method, this hierarchical method we've got, is a way of leveraging multiple sources of data, so Yeast-one-hybrid and time series expression. It performs well in silico experiments, and it also captures pre-existing knowledge from the literature. And finally, so this is kind of the main aim here, is that these models are useful for identifying specific regulations. So they could be condition specific, could be tissue, or they could be individual specific. But what we really want to do is we want to be moving on to cases where you have an orthologous species, and we want to know what the difference is. So why does this one look slightly more intelligent than this one? So I would just like to acknowledge a few people. So this is joint work with David Wild, Catherine Denby, and Vicky Buchanan-Wollaston, who are all at Warwick. And thank you to Zubin Garimani at Cambridge for some useful comments. Should acknowledge that this presentation was made possible by the ISCB Travel Fellowship, and the project that I'm working on, supported by BBS, this BBS SRC grant, and David Wild's supported by this SRC grant. That's it. Any questions?
Okay, so maybe I'll warm you up with one. In the previous talk, we saw a hairball.
Yep.
What are the technical issues in scaling up your method to that kind of dense network?
Yes, so I guess the main issue here is this normalization, where we have to normalize over this power set here. So even if you turn that into a truncated power set, they're still quite horrible, and we tend to limit it to things where there are hundreds of genes rather than thousands. So the limit here is really the number of regulators rather than the number of genes. So if you can get down to a handful of potential regulators, you can run it over thousands of genes.
Okay, Mukund?
Yeah, so it's a technical question about the Gaussian process as a prior. So how often do you find that you need to extrapolate beyond the range of inputs over which the measurements have been made? So the question is, how effective is it at interpolating versus extrapolating beyond the range of measurements, and how often do you actually confront that?
So as it stands within this talk, it's aimed more towards predicting network topology, so we haven't really had to do that. But we are actually using this to predict how our networks might evolve in terms of novel conditions. And we find that it recapitulates the data quite nicely, and it also recapitulates the data when you give it a subset training. But I don't know how well it does when you go to, when you go to completely novel situations. Again, it's going to depend on how good coverage you've got in your training sets.
Thanks.
Thank you.
Hi, I have a question about the inference. How are you doing the graph inference over the parents for each of the sort of the subgraphs that you have after you have your hyperparameters?
Yeah, so again, this goes back to here. So it's an explicit calculation over all truncated power sets. So if you've got, say 100 potential regulators, you can work out a truncated power set. So it could be gene 1 and 2, gene 1 and 2 and 3, gene 2, etc. And you have to explicitly evaluate a Gaussian process model over each of those.
So you explicitly calculate the marginal likelihood for every member of the power set. Is that
a truncated power set? Yeah.
Truncated power set.
Yeah.
So you go up to some KY's power set. Is that correct? Okay. Thank you.
So we still have some time. I might ask real quickly, what kind of computing resources did you need for the results you described today?
So, okay, yeah. So if you've got a small gene network, so if you're talking about 10s to 20 genes, you can probably run it on a desktop and it might take a couple of days or so. But once you start getting to hundreds of genes, you need a relatively large cluster. It's embarrassingly parallel, so you can run it, if you've got a big cluster, you can run it for lots of genes.
Okay. Well then, I think we should thank Christopher once again and talk to him offline.
Thank you.